Okay, we shall get started. Welcome back. You braved the first lecture and decided to come to the second. Congratulations. That's a win already right from the start. So do you guys have any, before we get started with the workshop today, do you have any questions from yesterday? Any things that you couldn't figure out? Allie said that she hosted a virtual office hours this morning. So any questions or Allie, any questions that would be worth my going over?
Mostly plotting questions, which we'll basically all cover tomorrow.
Okay. So mostly plotting, but we'll cover plotting tomorrow. Okay. Well, today is going to be a lecture about dplyr. Dplyr is a package, part of the Tidyverse, that is all things data cleaning, data manipulation, making new columns, performing calculations on new columns, etc. So it's one of the bread and butter core software packages that we will use in our workflows. And today there will be a lot less of me standing on my soapbox, convincing you that this is important, and much more of Akshay telling you about the dplyr package. So as soon as Akshay is done with tech support, we'll go ahead and get started. All right, come on up, floor is yours.
I thought you were going to go over.
No, it's all you.
Alright, so today we'll be introducing you to the dplyr package. The dplyr package enables you to take a data set and do things with it. Like you can choose rows or columns based on certain conditions. You can make new columns. You can rearrange your data based on certain conditions. And before we begin, I'd like you to go into folder number 2, so 2_dplyr, and open the dplyr student.rmd. So I already have it open. I'll close this one. So Matt introduced you to this kind of file. So this is an R Markdown file, and I'll sometimes refer to it, or I might always refer to it as an RMD file because it's saved as .rmd. And the way it's structured is you have chunks of code interspersed with just regular text. And the idea is to run the chunks of code in order. So the first thing you would do is when you see this is to run the first chunk. So that's your setup, which is the most important chunk. And let's go back to the talk. And for today's talk, we'll be using two data sets, proteins and mitocarta, as examples. And usually, so we'll show this to you on Thursday, how to take a real-life data set and import it into R. But for today's talk, the data sets we'll be using are in their own packages that Matt made. So the proteins data set just contains human protein information from UniProt, and the mitocarta contains data from the mitocarta project led by the Broad Institute. And yeah, these are the two packages. Now, if you go back to if you look at your RMD file, so this is the second chunk, Ali pointed out something yesterday that this package, this DevTools package, which is what we need to install Matt's packages, might not be up to date. So I just want to show you how to update a package. And to update a package, you basically just reinstall it. Matt showed you how to do that programmatically yesterday, but there are alternatives to that. So one way is to just go into Tools and click Install Packages. And in this space, you just type in DevTools. And the good thing about this is if your package doesn't exist in CRAN and you type it, it won't allow you to autocomplete. In this case, it does because DevTools exists in CRAN. And so just go DevTools and then you install it. And it'll give you this prompt. You go click Yes. I'll take a second to install.
So if you don't have the console open, you won't see the messages. You can minimize or maximize that window. But if you do have it open, then you can see restarting the session, installing packages, basically performing a couple of things. So it's nice sometimes to see when R is thinking or doing some work under the hood, so you don't sit there wondering when it's going to be done or what's happening.
Right. So yeah, if it were like this, we wouldn't know whether it was done or not. And so you just, oops, or make it extra big.
Actually, where's the what lib is unspecified
Does it say that?
at the top of the red?
Oh, lib is, I think that's because I didn't specify which library, I think, or where to install it. So R just decides for me where it wants to install the package. So that's kind of, it handles that. So unless it gives you an error message and says it wasn't able to install, I tend to ignore the message. So now that we all have DevTools installed, we need to open this package. So one way to do that is in this chunk that we haven't executed, you can just type in library, and Matt showed you this command yesterday. So this opens the package, and you leave everything else as is. And once you've done that, you can just hit play, this play button here, and it should just run through each line. And it did it fine on mine. It should hopefully do it fine on everyone's. And now you should have both the proteins and the mitocarta datasets for this talk.
So that's a good point. You need to make sure to load your library DevTools above the line that calls DevTools the package. So Akshay put it in the right spot, but just make sure that you don't put it below. Otherwise, it will be looking for a package or a function, which is install_GitHub. That's the function that we're using from the DevTools package, and it won't find it. So make sure you sort of load this first, and then you can go ahead and call those functions.
All right, if everyone's done that, so before we discuss the various verbs that are in the dplyr package, let's get a sense of what kind of data sets, proteins, what kind of data proteins and mitocarta contain. So one really simple function is this dim function, stands for dimensions. And if you execute the chunk that you see, the dim and of the proteins data set, it just gives you two numbers, because this is a rectangular data set, and in matrix notation, the first number refers to the number of rows your data set has, and the second number refers to the number of columns. So this has 20,430 rows and 8 columns. In the tidyverse, rows are frequently referred to as observations, and columns are frequently referred to as variables. So I might use those two terms interchangeably also. The dim function is a very simple function that gives you a very simple output. More sophisticated function to get a sense of what data you have is to use the glimpse function that exists within the tidyverse. And the syntax is the same as for dim, except instead of dim, you have glimpse. And if you run that code, you'll get this output, which is a lot more comprehensive than what you saw previously. In the previous example, you just saw two numbers, the 20,000 and something and 8. In this case, it explicitly tells you that the 20,430 refers to the number of observations, or rows, and the 8 refers to the number of variables, or columns. And it also tells you what those variables are. And it gives you the first few entries for each of the variables, and it tells you what kind of variable each is. So in this case, that's a character variable. In this case, that's a double. And we'll go over what the different these different types are. Matt showed you the dollar symbol, which is frequently used in R. In this case, they have it here. So what this means is if you wanted to extract the values of, for example, uniprot_ID, you would just type in proteins, $uniprot_ID. So that's what that $ there means. And the two pound symbols just signify a new line, lines. I should do that too. So if I go down, execute glimpse, didn't work. Oh, it worked. I get the same output. So in R, there are 6 basic data types. There's a character, which we saw earlier, the CHR. So in this case, it's like, for example, the letter A or the word tidyverse. There are numerics, which are either whole numbers like 2 or decimal numbers with decimals like 11.5. They're integers, so this is a way of explicitly telling R that this number is an integer. So you'll often see in your global environment this big L, and that lets R know that that variable, for example, only contains integers. So example, in this case, it's the integer 2L. Then there's a logical, which is either true or false, it can only take one of two values. And then there are complex numbers, so a real number plus an imaginary number. And finally, there's something called a raw data type, which I still don't know what that is, but it exists in R. You'll also come across the double data type, or DBL, and for historical reasons, this naming exists, but it's basically, when you see double, think numeric. So numeric and double, they're interchangeable. Finally, there are these things called factors, and this is a really important concept. So you can have categorical variables that are, or character variables that are unordered, like the names of 3 people. They don't necessarily have to have an order, but the names of the months do, January, February, and so on. So if you were to enter the names of the months into R, it would, by default, think that the first month is April, because it's going alphabetically. So you have to then tell it what the correct order is. And this makes a big difference in, for example, when you're trying to plot graphs and you don't get like bars in the right order. Frequently it's because it's a factor issue. If you wanted to see what kind of variable a specific variable in your dataset is, you can use the class function. So in this case, I told you about the $sign, so that's a way of extracting a specific variable. So if I wanted to see what type of variable length was in my proteins dataset, I would run this class, and in brackets, proteins $sign length. And the output is, it's a numeric, which makes sense. So if I execute that chunk here, I get the same output. I'd like to reintroduce the...
Can you explain what the code before that used?
Which code?
Sorry. Before the thing that you're executing, the glimpse echo.
Oh, okay. I think Matt's going to introduce that also. But so the echo equals true. In this case, it's telling when you knit this file into and I did it into an HTML file, it'll show you the code also. If you set it to false, it'll just show you the output. So in this case, you'll just see numeric in your HTML document. You won't see the code. So this is a nice way of, like, if you wanted to make a PDF file for someone, but they weren't interested in the underlying code, you could set it to echo equals false. But since I want to show you both the code and the output, I've set echo equal to true. And similarly, I said warning equal to false, because sometimes R will give you warnings, and it's a similar kind of thing. That might be useful to you as the person writing the code, but for someone reading it, they might not be interested in what warnings you got. So
We have a whole lecture on Friday about that, so that will become clear. Just wait a couple days.
So this is the pipe operator, and it's a way, as Matt said, it's a way of chaining together lines of code in a way that makes it easy to understand what you're doing, and also very easy to understand what someone else has done. And as an example of how chaining works, in this case, you take your proteins data set. As Matt said, when you see this pipe operator, you always think and then. So you take your proteins data set, and then you select 2 columns, in this case, uniprot ID and length. And then you filter out you filter in, I guess, all the proteins whose length is greater than 500 amino acids. And in my case, the only difference between this and what you see in your chunk is I've put a little head operator. So this is a nice function that takes an input and gives you the number of rows that you've specified. So in this case, I've specified 1. And the only reason I've done it is for the purpose of this talk, because if I didn't do that, it would try to print out the entire output. But in this case, it just prints out 1 row for me. But in your case, if you go to the R Markdown file and execute that chunk, you see it outputs everything. In this case, it has 8,000 rows. And you can see you selected only 2 columns, so your output is only 2 columns, and you filtered all the proteins that were greater than 500 amino acids. And if you look at the numbers there, it disappeared. They'll all be above 500. So yeah, as Matt said, when you see the pipe operator, you just think and then. So a handy shortcut, so it's kind of tedious to keep typing in percentage greater than percentage, is to go, if you have a Mac, it's Command-Shift-M, and if you have a Windows operating system, it's Control-Shift-M. So the alternative to using this pipe is to just do everything in one line, which some people might argue is a more efficient way of doing it, because now I have 1 line of code as opposed to 3. But the problem, and if you execute that in your RMD file, you should see that it gives you the same output. But the problem with this is it's, at least for me, less intuitive to understand what it is I'm doing, and it's also more error prone, because if this gets more sophisticated, the coding you do, you can oftentimes get multiple brackets, and it's very easy to lose track of that and delete a bracket, and then you just get errors that are difficult to debug. Okay, so as I mentioned at the start of the talk, the dplyr package allows you to do a number of useful things with a given data set. So some of the things you can do, you can select rows and columns that match certain criteria that you're interested in. You can create new variables. So Matt showed, gave you an example of the mutate function. That's a way of doing that. And you can obtain summary statistics for the whole data set, but where it gets very powerful is you can do it by group. So an example of that is if you had a proteomics data set, you could work out the average protein length, or you could work out, if you had the appropriate variable, you could work out the average protein length per organelle, which might be useful. So the main function, so dplyr is a very big package with many, many functions, but we will be covering these verbs in today's talk: select, filter, arrange, mutate, and summarize, and finally group by. And group by and summarize work together really nicely. So the first verb is select, and so within the tidyverse, when you say select, you always are referring to columns. And when you say filter, you're always referring to rows. So I might say select rows, but I mean that in the colloquial English, the standard English sense. So the most basic select function is to just take in one or more column names. So if you have more than one column, you would separate them by commas. And a simple example is this. You take your proteins data set, and then you select 2 columns that I've specified here, uniprot, ID, and length. And once again, I've piped it into this head function just so that you see one row as opposed to, in your case, all the output rows. So you get this. And I'll run it again, and what's that? Now, if you wanted to select everything except a specific variable, you could, this is the syntax you use. You take the name of that variable and you put a minus sign before it. So in this case, I want all the variables in proteins except uniprot ID. So I take proteins and then basically unselect uniprot ID. And you see the output here, the proteins data set has 8 variables, but because I've now removed the uniprot ID variable, it now you can see has a 1 times 7 variables. So slight detour, I forgot to mention there's this thing called a tibble, and that's a made-up word that only exists in the tidyverse. And a tibble is basically a data frame that is slightly different. And I can show you here. Like for example, R has a built-in data set, has a range of built-in data sets. One of them is called iris, which has information on the iris flower. Now, if I were to just run it, so that's just a regular data frame, you see it's given me all the values. And you have no idea, I have no idea what the first few columns of numbers mean. But if I convert iris into a tibble, so I can do this as.tibble function, and I just move up a little bit, it cleverly just gives me the first 10 rows. And so then I can see what the columns mean. So it doesn't just show me everything. It shows me things that fit nicely in my console, so I can see everything. And then it also tells me there are 140 other rows. So that's really the only difference. That and if there were any negative values here, they would pop up as red. So it's just an aesthetic difference. So if you wanted to select multiple columns, let's say you wanted to select 50 columns that are in a row, instead of typing them all out by name, which is tedious and error prone, you can put you can put a colon between the first variable and the last variable. So in this case, this chunk of code, you take your proteins data set and then you select uniprot ID and protein underscore name and everything in between it, in between those two. So in this example, the two columns that are in between these two bookending columns are gene name and gene name alt. We run that here. You see it gives you 4 columns and all the rows. So in your R Markdown file, if you can't see the other columns, you can just click on this arrow here, and it allows you to scroll to the right. Cool. All right. We'll just take a couple of minutes to just do an exercise. So I'd like you to take the proteins data set and select these columns that I've named here. So these 4 columns. And as Matt said yesterday, copy pasting is fine.
Akshay, should we do this at the end of the R Markdown file?
No, but so there's, I should say, there's an empty chunk right under the this exercise.
Okay.
So if you do it within that,
Oh,
and then you
got
just
it,
execute
got
that,
it.
then it should be there. Right. Okay.
So I just want to interject something before Akshay continues on. So this concept of the pipe, one thing I want to be explicit about and mention to you guys is that we string these commands together, but what ends up happening is that whatever you generate from the previous line of code gets carried to the next line of code. So for example, if you just type select and then the columns that you want to select, if you didn't type proteins and then select, select doesn't have anything to select on, right? So what you can think about is if you generate something in the first line and then you pass it to the second line, you can do something to that code in the second line. And let's say you generate a new column, and then in the third line, you can do something to that new column. Now, if you switched row 2 and 3, you can't do something to a column you haven't generated yet. So this is all very sequential. So keep in mind that when you're going through this and then type of literate programming, what you want to keep in mind is that because you'll run into errors, you'll run into bugs, you'll see, okay, this doesn't, it can't find this or it can't find that. Just remember that you want to think about what is carried from one line of code to the next line of code to the next line of code and so on. So I just wanted to be explicit about this and then where you're piping these functions, but remember the functions are generating something, they're taking something, right? This is an object, right? The object is proteins. In this case, your object happens to be a data frame. And then you do something to that object and carry it forward. Now, if you selected these and then you did something in the next line of code that was not on one of these columns, you would have an error, right? Because that column is not propagated forward because you selected these 4. So just something to keep in mind.
All right, so this is an example of a solution. So you could have typed out all the variable names, so there are 4. But in this case, sequence length and mass are 3 consecutive variables. So you can use a colon to get all 3 of them in. So is everyone clear on just the intuition of select? So when you do select, you just are focused on variables that you're choosing. And as Matt said, once you've done a select, so let's say your original data frame consisted of 8 variables and you select 4, and then if you're continuing piping in that code chunk and you want your 8th variable that you selected out, it'll give you an error because you no longer have that, you've selected out that variable. So one thing you can do when you have, so in this case it's just 2 lines of code, but let's say you had 8 lines of code, is you can comment out, you can just execute the first couple of lines and look at your output, then the next 3 lines, then the next 4 lines, and see how your output changes, just to have a sense of what is happening, and also just to make sure that you're not making any mistakes. So just as select applies only to variables and or columns, filter applies only to rows or observations. So you can filter in or out rows based on certain on logical statements. So you have a certain logical statement like, is this equal to, is this not equal to, is this greater than or lesser than? And the rows that for which that statement is true are kept, and the rows for which that statement is false are discarded. So a really simple example is, so you can filter both on categorical data and on numerical data. So a simple example of a categorical filter is this. So in this example, we're using the mitocarta dataset. So this code, you take your mitocarta dataset and then you filter only those proteins that have a mitodomain score that is equal to mitodomain. So this variable, mitodomain underscore domain underscore score, I think it can take four. It can contain one of four conditions. So it's mitodomain, no mitodomain, shared domain, and just NA. So let's say you're only interested in those proteins that have this mitodomain. This is something you could do to just get those proteins. So once again, so this output, I only have one row because I'm using that head function. You'll have all the output rows. Since this is quite a rich data set, so it has 43 variables or columns, in this, on this screen, you'll just see a few of the variables with their rows, in this case just one. And once it hits the end of the page, it just says, there's no space to show you everything else. So in this case, it just says, there are 37 more variables, and it tells you what all the variables are. But in your R Markdown file, when you execute that chunk of code, you will see everything. So you can see it has 426 rows. So there are 426 proteins in this data set that have mitodomain. And you can just scroll through to see all the variables.
So why do you have two equations?
Oh, that's a good point. I meant to say. So in this case, those two equals. Sorry. Okay, those two equal signs, that's an equality. It's asking R if a specific row, if for that row, if the mitodomain score is equal to mitodomain. If you just did one equal sign, which I do a lot, that's you telling R to assign something to something. So Matt showed you yesterday, that's one way of, like, if you do B and then arrow 2, you're assigning the number 2 to B. But you could just do B equals 2. So that's why it's a double equal. Thanks, Matt. I'll just make sure not to touch the cables. All right, so that's a way of filtering based on categorical data. And you can filter on numerical data. So it's a similar kind of operation you perform. So in this case, let's say, so there's a variable mcarta2 underscore FDR. So let's say you had particularly stringent conditions and you only wanted those proteins that have an FDR that's less than 0.05. You would run this chunk. So you take your mitocarta dataset and then you filter in only those proteins that have an FDR less than 0.05. So for all the rows for which this condition holds true, you will then get that as an output, and all the ones for which it's false get discarded. So again, similar output. And if we run the code chunk here, there are 947 proteins for which this is true. And again, since you're doing a filter function, it doesn't affect your variables. So all the variables that are in the original data set will be in your output. Now, if you wanted to filter on multiple conditions, you can just string together a bunch of filter commands. So in this case, I wanted to, I almost said select. I want to choose rows for which the mitodomain score is equal to mitodomain and the FDR is less than 0.05. I would just, again, take the mitocarta dataset, pipe it into filter, the first filter command, and then take the output of that and filter it into the second filter command, and then see the output. So if we execute that here, you see there are now only 328 proteins for which both these conditions are true. Yeah.
So, so you had previously mentioned you use sort of like factors and characters
Yeah.
as object types. What what are the, I guess, what's the difference between having a variable that's a character as opposed to a factor? So for instance, it seems like mitodomain score would be a good variable to have as a factor.
Right.
But
So
yeah, I guess what,
in this case, I don't think there's a difference. But if you were to then plot that, and you wanted your bars to be in a particular order, then you would have to change, if you wanted to. You could change the order of, explicitly change the order of that factor. But yeah, thanks for that. So these were only two filter commands.
I'll also just add, it'll become more apparent how to leverage factors tomorrow when we start plotting, because a factor is something that you can take advantage of in plotting, whereas your character is just a type. It'll become more apparent tomorrow, so ask it again tomorrow.
So in this case, we just had two filter commands, but let's say you wanted 20 filter commands. Instead of writing them all out, which again can be error prone and takes up a lot of space, what you can do is you can just use one filter command and then put in all of your conditions within that one filter command and just separate them by commas. So in this case, the comma means and. So I want proteins for which the mitodomain score is equal to mitodomain and the FDR is less than 0.05. So this is identical to the previous chunk of code, except I have one filter command instead of two. We execute this, we get this. We do it in the R markdown. And once again, just as a sanity check, previously there were 328 rows. There are 328 rows again, so 328 proteins. All right. So for your filter exercise, I'd like you to take the mitocarta dataset and choose those proteins that have a mitodomain score not equal to mitodomain and an FDR that's greater than 0.05. And just as help, the equal to sign, the equality is equal equal. Here, if you want to say not equal to, you would do exclamation mark equal. I don't know why I have this here, but if you wanted to do less than, it would be less than and then equal. And if it's greater than and equal to, it would be greater than and equal. All right, so if everyone's okay, I can reveal the solution. So this is one solution. You can use just one filter command and put in both the conditions separated by a comma, or if you wanted to be more explicit about it, you could have two consecutive filter commands. So that's select, which applies only to columns, and filter, which applies only to rows. The next verb within the dplyr package we'll be discussing is the arrange verb or function. So arrange is a really useful function for changing the order of the rows. You could, for example, rearrange the rows so that they go from lowest to highest of a certain numeric column, like for example, from the shortest to the longest protein, if you wanted. By default, when you apply a range to a numeric variable, it goes in ascending order. So, for example, again, going back to now the proteins dataset, if you take your proteins dataset and then apply the arrange function to the length variable, what it does is it takes the length column and will now rearrange your entire dataset so that you now have your shortest protein as your first row and your longest protein as your last row. So if we execute that chunk here, and if you just move to the length variable, you can see it starts off with a protein that has just 2 amino acids, and it goes up. So this is a nice way of, for example, if you quickly wanted to find out what the 10 shortest proteins in your data set were, you could do something like this. Conversely, if you wanted to see what the 10 longest proteins were, you need to use a descending function within your arrange function. So this is identical to the previous code chunk, except within the arrange function, you can see there's a descend function. So you take your proteins data set, and then you arrange. Again, you're arranging on the length variable, but you're specifying that you want it in descending order. So we run this chunk, and then once again, oops, it's taking a little while. Once again, if we move along to the length variable, you now see it's the longest one, and then the second longest, and so on. Now, just as so this is the arrange exercise is just to think about how arrange would apply to a categorical variable. If you would apply it to a character variable, what do you think would happen? How it would arrange the rows here?
Sorry, what does it mean if in a chunk there's the little play button is not present?
Oh, I have not seen that.
Anybody else see that?
Is anyone else having that play button issue?
Don't worry about it. Go ahead.
So if I were to apply this to a categorical variable, how do you think it would rearrange the rows? Alphabetically. So as an example of a character variable, I use the gene name alt, so that's the alternative gene name. And in this case, if you look at the output, so gene name alt, in R, if you put in numbers, it goes 0, 1, 2, 3 to 9, and then A, B, C, D. So that's why that's the alphabetical reordering here. So if you just had gene names that started with letters as opposed to numbers, it would be very clear the order in which the rows were arranged. So that's it. So arrange is a very simple function. You can either arrange ascending or descending, numerically or alphabetically. Yeah.
Did you do the same thing with arrange, descend?
Yes. Yep. Yes, that's a good point. So the arrange descend applies to both numeric and categorical variables, correct? So the three functions we've gone over so far within the dplyr package all they don't generate new variables. The mutate function is special because it's the first verb in this talk that does that. So Matt showed you a couple of examples yesterday. So what the mutate verb does is you can take existing variables and manipulate them in some way to create a new variable that contains the output of whatever it is you did. So in this case, again, we're using the proteins data set. What this code chunk does is it takes the proteins data set and then it calculates the square root of the length variable. So it's working out the square root of the protein length for each row, so for each protein, and is taking the outcome of that operation and populating a new variable or a new column, in this case square root underscore length, with those values. And this is the syntax. It's always the new one you want to make on the left equals to some operation you apply to an existing variable or variables. In this case, it's just one, length. And the name you give to the new variable is whatever you decide. So in this case, I thought square root underscore length was helpful, but you can call it anything you want. And again, I've piped it into a head function that will just show me the first row. Oops. So if you run that chunk, and another thing is when you make a new variable using the mutate function, by default, R will make that column the very last one in your data frame. So if you scroll all the way across to the end, you see there's a new column called square root length, and all those numbers are the square roots of the corresponding numbers in the length column. So 495 square root is 22.2 something, and so on. Now, that was an example of taking just one variable, doing something to it, and creating a new variable. You can take as many existing variables and use them to create a new variable. So in this case, you take the proteins data set and you use the mutate function to create a new variable that I've called protein underscore length underscore mass. And that contains the values of the length of the protein divided by the mass of the protein for each protein. So in this case, if we execute that chunk, we get this output. And once again, the new column appears at the end. And you can see it's a new column called protein underscore length underscore mass. And it has all these values that are equal to the length of the protein divided by the mass of the protein. And you can see they're very small values because the number is much bigger than the length number. All right, if that's clear, then as an exercise, I'd like you to, again, use the proteins data set and create a new column, give it whatever name you like, and fill it with protein lengths divided by 100. And as far as naming convention goes, try to use lowercase and don't use spaces. Although you can still do that, it's just nicer to do underscores. And try to avoid, like if you're calculating the mean value, try to avoid creating a new variable name called mean, because there's a function called mean, and things can get confusing. Yeah?
You put a number in the name, is that going to screw it up?
I don't think so, because it'll just call it a character. Yeah.
Yeah, for those interested, there's an entire style guide to the Tidyverse that makes suggestions about how to name things and good conventions and what code should look like in these things. That's for next week's boot camp. For this week, we'll just focus on teaching you some of these fundamentals. If you do something and it breaks, let us know. But it's a pretty flexible platform, so you should have a lot of leeway to do with it what you would like.
All right, so if everyone's okay, I'll reveal the solution. So again, syntax is really simple. You take your proteins data set, you pipe it into this mutate function, and your new variable, which in this case I called protein underscore length underscore 100, is equal to what you're supposed to do. You take your length variable and you divide it by 100. So then what this does is it takes each row, takes a value in the length column, divides it by 100, takes that resulting value, and puts it in the corresponding, I guess, cell within your new column, protein underscore length underscore 100. So is the mutate function clear? The intuition? Cool. All right, so moving on to the summarise function. So what summarise does is it, as the name suggests, it basically aggregates across all the values of a given column based on a certain condition. So for example, if you wanted to work out the mean protein length and mass, you would execute something like this. So again, we're using the proteins data set for this example. You pipe it into the summarise function, and there's a mean function within base R. So you'd use the mean function to calculate the mean of the length variable and the mean of the mass variable. And also, just as an aside, I use the S because I'm Indian, but you can use a Z, and that's fine. And the same thing with color. I spell color with a U, but you can spell it without a U, and it's fine in the tidyverse. So the output it looks like this. And in this case, I didn't use a head function because the output is just one row that has two values. One is the mean length of all the proteins in the proteins data set. So it turns out the mean length is 557 amino acids, and the mean mass. And yeah, it's a very simple function. If you wanted to give the columns, so in this case, it's calling each column this mean in brackets length and mean in brackets mass. If you wanted to give them prettier names, you can explicitly define those names. So in the previous example, I just piped in proteins into the summarise function, and I just did mean length, comma, mass, mean mass. In this case, I'm saying that I want the column names to be mean underscore length and mean underscore mass. So the output is identical to the previous one, except my column names look nicer. And I have an example later on of using summarise in tandem with group by that really makes clear how useful the summarise function is. But this is an example of a simple use of this function. So as an exercise, there's not that much to the summarise function. I'd like you to make a new table that contains the mean, median, and standard deviations of all the proteins in the proteins data set, of the lengths of all the proteins in the proteins data set. And just as a hint, just like you had the mean function to calculate mean, R has a median function to calculate median, and SD to calculate standard deviation. Okay, so if everyone's okay, if everyone's done with that, this is a possible solution. You take the proteins data set and then you summarize the mean length, median length, and the SD, standard deviation of the length. And in this case, I've explicitly defined the names of the output columns, but you could do it without that. You could just do mean length, median, and SD, and that would be fine. And the output, it looks like this. So that's summarize. And the final verb we'll be covering is group by. And as I said previously, group by and summarize work really nicely in tandem. So in this case, if in this example, I'm using the mitocarta dataset to work out the mean length of proteins that are both mitochondrial and non-mitochondrial. So the mitocarta dataset has this variable called mcarta2_list, which contains one of two values, 0 or 1. If it contains a 1, that means the protein is mitochondrial. If it contains a 0, that means the protein is not mitochondrial. So in this case, what you're doing is you take the mitocarta dataset and then you group by. You're telling to group by the mcarta2 list variable. So subsequent to this, R is going to perform whatever operation you want later on separately on both the non-mitochondrial, so equals 0, and the mitochondrial proteins, the ones that equal 1. In this case, I wanted to work out what the mean protein length was for mitochondrial and non-mitochondrial proteins. So just as in the previous case, I use a summarize function, and then I work out the mean. Now, if I were to skip the second line, just go mitocarta, pipe it into summarize, I would get the mean protein length of all the proteins in the dataset. By using this group by on this _list variable, I will get two outcomes, mean of mitochondrial and mean of non-mitochondrial. So this is what my output looks like. So the proteins that are non-mitochondrial, so for which mcarta2_list equals 0, the mean protein length is 590 amino acids. The ones for which it's equal to 1, so are mitochondrial, the mean length is 400 amino acids. So once again, I haven't used a head function here because the output is just a couple of lines. So yeah, I use group by a lot for this kind of operation.
And just to mention a convention, in R, as in with several programming languages, 0 and 1 oftentimes refer to false and true. So it can be a numeric, but in this case, what the data is referring to is 0 is false, meaning it's not included in the mitocarta list, or 1 is true, it is. So it's not relevant for this example. We can group by just about anything, but just to let you know that that's a convention that you'll see sometimes.
And also in R, if something is evaluates to true, it's automatically given a value of 1. And if it evaluates to false, it's given a value of 0. So if you take, I don't know, a data frame of 20,000 rows, and 1,000 of them evaluate to true on whatever condition you've selected, if you use the sum function, it'll give you 1,000. So you know, okay, 1,000 of these rows evaluates to true. So that's a nice way of getting a sense of how many rows in your data set are true for whatever condition you've supplied. So that's group by. So far, everything we've done has generated a temporary data frame, a temporary output. Presumably, when you do your own analysis, you would like to, I'm guessing, save the eventual data set, but also save intermediate ones. And to do that, it's really simple. You can use a left assign or a right assign operator, or you could even use an equal sign, but our convention is to use particularly this left assign operator. So this is an example of assigning your output. So the code on the right of the operator, that's what we just ran, but we didn't save the outcome, the output. To save the output, you just do this left assign operator at the very top and into an object that you've created. And you can, once again, give it whatever name you like. I've just decided to call it mito_new. And when you execute this, you will see, whenever you save something, you will see on the top right hand, so right now your global environment is empty because we have, so far we haven't saved anything. But if I run this chunk, you see it's now saved it. And one thing that's nice to do is if you click on it, you get this kind of Excel type output. So this is really easy to, it would be more relevant if this were a bigger data set, but you could do things like you have a little search box, you can filter, you can do various things that you'd be like, the same things you can do in Excel in a table format. But that's just saving the data set in your global environment. If you then exit R and come back in, it's probably not going to be there. So if you want to save it as a file that you can then send to someone or just save on your computer, that's really easy to do. So in this case, if you wanted to save it as a .csv, you could do anything. You could do .csv, .tsv, .xls. In this example, if you wanted to save it as a CSV file, you can use this function write_CSV. And on Thursday, I'll be discussing different functions that you use to import. These are the analogs to those functions. So instead of importing something, you save something. So it's kind of like exporting it onto your hard drive. And the format here is the function is write_CSV. And the first argument it takes is whatever data frame you want to save. So in this case, I saved it as mito_new. So that's what I have to put in there, comma, the name of the CSV file. And again, I can call it whatever I want, but it has to be end with .csv. And once you do this, it will save this data frame as a CSV file in your working directory. So it's really important to know what your working directory is. And I don't think we'll cover this, but this is why it's good to have what are known as projects in R, because whenever you do something within a certain project, whatever you saved will be saved within that project's working directory. So you can toggle between projects and be confident in knowing where all the things you saved are now in. So that's pretty much it.
If you want to know what your working directory is,
Yes.
you can use a little function in R. And it's just get, I think it's underscore.
Yeah,
And parentheses. And it will tell you your working directory.
sorry, Matt. So I
Get WD.
have typed in get WD, open brackets, close brackets, and if you type that out in your console and hit enter, you will see which directory. If you lose track of where you are, or like where the stuff you've saved goes to, that tells you where it goes, where it's gone. And finally, lots of our packages, especially the packages within the Tidyverse, have really helpful help files that are known as vignettes. So these are just guides. And you can just Google, Tidyverse guide to dplyr or dplyr vignette. But one way to do it within R is to use a browse vignettes function, which I believe is in the very last chunk. So if you execute that, it sends you to the help documentation for the dplyr package. Those help files are really useful because they are thorough. They thoroughly each function, and in this case, for each function, I use just one, maybe two arguments. I think I just use one argument for each function. But each function can take on a whole list of arguments. So it goes into all of that, and it also gives you really helpful examples. Right.
Are you getting a 404 if you click on any of those?
That's what we got last time.
Yeah, so that's a problem with trying to access this through RStudio Cloud. Another way that you can access these is if you go over to here to the help and you type in dplyr. You can come down and click index at the bottom, and this is going to be everything within the dplyr package. One of the links here is to get to the vignettes, and here are actually several vignettes about dplyr. So you should be able to click on any of these, and then you can read the vignette right there. So that 404 error is just a problem with the way that RStudio Cloud is interacting with RStudio, but the information is still all within the package. So the vignette goes along with the package, and we have the package installed, so it's all accessible.
Any questions?
So you introduced the arrange function
Mhm.
to arrange sort of rows. Is there an easy way to arrange columns? Say my PI wants, the G name or whatever that's like way over here at the very front or something.
What you can do is you can use the select function to rearrange the rows by just selecting the rows in the order you want them in. That's one way to do it.
Yeah, so if we just go view mitocarta, and we look at the last column here is this here, HPA. So let's say that you're overly demanding, cantankerous, and unreasonable PI wanted to see that first. You could do something like mitocarta, and then select, and what you would do is HPA this, right? So it selects that one, and then what you can do is essentially take the rest of them. So you can go training data set all the way to test this total peak area. Remember the colon is going to be everything through. So then that puts this in the first position, something like that. Yep. Any other questions?
So if you want to stay back for a few minutes, there's another RMD file within this folder. It's called dplyr workshop, and that has a bunch of questions. And it's using the chromosome dataset, which is within the Tidybiology package. And there's one question where I ask you to calculate the total number of RNA, because there's like microRNA, non-coding RNA, different RNA species. And the last time I did this, someone said that my proposed solution was inelegant. So that's an extra challenge. It took me a little while to work out what the elegant version was. So if you could do both, but that's using functions that we haven't covered in this talk. But yeah, so
well, it's also a good opportunity to mention, right? And Akshay has done a good job explicitly saying on at least three occasions, this is one solution to the problem, right? Because like with the filter, you can take your data set and then filter a row and then filter another row. That's one solution. Or you can filter one argument, comma, another argument, and so on. Another solution is nested, right? Like nested works. So the point is a little bit like the point I was trying to make with the programming language. It doesn't matter which language you use. What matters is how you're able to accomplish the thing that you want to accomplish. And again, there are style guides and suggestions and these sorts of things, but for the most part, what we want to do is give you tools, show you there's a couple of different ways, but really try to embrace this mindset. There's no right single way to do it. There's a way to try to accomplish a task, and there are several ways to get there.
And as a final point, I just noticed, so the mitocarta dataset here is a tibble. And you can see, so I said previously, if you have negative values, they come up as red. I'm just reminded, so does NA, so not available. So this is really cool. If you wanted, for example, if you were interested in HBA primary, whatever that is, you wouldn't be interested in 7 of the first 8, because they're all NAs. And this makes it very clear. But if this were a data frame, they'd all be black. So that's another minor difference. Minor but useful. Yeah.
So in terms of that, what if you want to put all the NAs up front, like in the first, I don't know how many rows?
I wonder if you arrange if it does NA first.
No,
Does it not?
All the way back.
Yeah, I think actually it's regardless of how you arrange, either descending or not, I think the NAs end up at the bottom.
They end up at the bottom? Okay.
Yeah.
Okay, I know that.
Yeah. So there is a argument that you can add to a filter is .na, where you can then say I want things that include an NA, or I want like a subset that is not NA. So you can do like the exclamation point is .na. So there's special ways to then summarize your NAs to get a sense of like missing values among your data. but that's good to know that it's there. We won't spend time going over it. Other questions? All right, in the last 15 minutes then, go ahead and take some time. We'll all sort of meander around, try to help you finish up some of the assignments. And tomorrow we will be back same time for Ali giving a talk on plotting and data visualization.